Hello students, this is Dr. Faizan Mirza. We are discussing gas transport by blood. When we are discussing gas transport by blood, the main gas that we are concerned with are oxygen and carbon dioxide. So starting off our discussion with oxygen. Oxygen is dissolved in blood, in red blood cells as well as in plasma. 2% of oxygen dissolved in plasma and 98% of oxygen binds to hemoglobin to form HbO8, which is oxygenated blood. Here, oxygen binds to the iron porphyrin ring. There are 4 iron ions present in one hemoglobin molecule. So 98% of oxygen is carried in that way. Normal arterial blood with a PO2 of 100 millimeters of mercury will have 0.3 ml of oxygen per 100 ml of blood. What is oxygen capacity? It is the maximum oxygen that can be combined with hemoglobin. We know that 1 gram of pure hemoglobin can bind 1.39 ml of oxygen. So 15 gram of hemoglobin per 100 ml is having the oxygen capacity of 20.8 ml oxygen per 100 ml of arterial blood. Bohr's effect actually tells you how oxygenated blood actually delivers oxygen to the tissues. And this delivery is not just dependent on how much oxygen is there left in the tissue, but also on the carbon dioxide present in the tissue. So the delivery of oxygen to a tissue depend on 2 factors. Number 1, the diffusion gradient which exists in that tissue, which means PO2 of the tissue, and the PCO2 of tissue, which means what amount of oxygen is already left in the tissue and how much carbon dioxide is accumulated in that tissue. Tissues with a higher PCO2, which means they have more carbon dioxide dissolved or more carbon dioxide produced, they receive more oxygen from the oxygenated blood. So blood passing through such a tissue which is respiring at a high rate will have more oxygen being delivered. H plus ions will enter red blood cell. They will combine with hemoglobin to form HHB, the hemoglobinic acid. This is what I will be discussing here. So let's suppose this is your tissue here. These are the red blood cell, and this is the plasma. This is the capillary endothelium. This is the tissue fluid here. This tissue is having a low oxygen. Why? Because it has respired all the oxygen. The blood that has come, it's having hemoglobin with oxygenated blood. So one hemoglobin is actually having 4 oxygens combined to it. So you can see there is a diffusion gradient, high oxygen here and low oxygen here. So we know that down the diffusion gradient, oxygen will move from the red blood cell into the tissue. This is because of the diffusion gradient, which I mentioned the first point here, why oxygen is delivered to the tissue. But the second point here, this tissue is having a high CO2 because the tissue was respiring at a very fast rate, so it had accumulated a high carbon dioxide concentration. This high carbon dioxide concentration will start to move into the red blood cell, again down the diffusion gradient. The carbon dioxide that moves into the red blood cell combines with water, resulting in the formation of H2CO3. H2CO3 then dissociates to form H plus and HCO3 minus ion. HCO3 minus ion leaves the red blood cell, and this negative ion is exchanged for chloride. So the chloride ion move from plasma into the red blood cell. This is called as a chloride shift. The H plus ion then binds with hemoglobin to form HHB, which is hemoglobinic acid, which I mentioned here. So the hemoglobinic acid forms, and the formation of hemoglobin causes more oxygen to be lost from the hemoglobin to the tissue. This is called as the Bohr's effect. So as the result of HHB formation, the tissue with a higher carbon dioxide concentration ended up receiving more oxygen than only diffusion gradient would have provided to it. The pH of blood, we know it's 7.35 to 7.45. It's mainly alkaline, slightly alkaline. It is basically alkaline because of this bicarbonate ion, which is present in the red blood, which is produced in the red blood cell and then enter into the plasma, and that's where it travels. So just discussing this thing again here. So you have the tissue, the respiring tissue here, and this tissue is having a high PCO2, and this tissue is having a low oxygen supply, low oxygen concentration at the moment, not the supply, low oxygen concentration here, and the red blood cell came with a high oxygenated blood. So this hemoglobin bound to oxygen will deliver oxygen here. Carbon dioxide will move into the red blood cell. It forms H2CO3 by combining with water. H2CO3 dissociates to form H plus ion and bicarbonate ion. Bicarbonate ion goes into the plasma, and chloride ion enters into the red blood cell. This is called as the chloride shift. The H plus ion then causes HHB formation, and oxygen dissociates from the hemoglobin. As a result, the blood ends up giving more oxygen to this tissue which was actively respiring. This was called Bohr's effect. The reverse of Bohr's effect occurs in the alveoli, where in alveoli, where the red blood cells go, these red blood cells are carrying hemoglobinic acid, and the alveoli is rich in oxygen. So you know that here the diffusion gradient will be that a high oxygen in the alveoli and a low oxygen in red blood cell will cause the oxygen to move from the alveoli into the red blood cell, and a high CO2 in the red blood cell and the plasma will cause the carbon dioxide to move from these regions in the blood to the alveoli down the diffusion gradient. So how do we begin this process? First of all, the bicarbonate ion which was traveling the plasma, it enters into the red blood cell. The movement of the bicarbonate ion into the red blood cell caused the chloride ion which was in the red blood cell to move out back into the plasma. This is called as reverse chloride shift. The bicarbonate ion then combined with H plus ion from HHB and forms H2CO3, whereas the hemoglobin is then left to oxygen to bind to. The H2CO3 dissociates to water and water and carbon dioxide. Water and carbon dioxide then come into the alveoli, and when they come into the alveoli, we exhale this thing, these two together. This entire process, which actually is completely reverse of Bohr's effect, is called as the Haldane's effect. The graphs here are three curves. These curves are shown for hemoglobin dissociation, and if you look at the axis, this is PO2, which means you are increasing the oxygen around the hemoglobin to this value, and you are increasing what increases alongside is the concentration of saturated hemoglobin. So at about this much of oxygen supply to the blood, you see that almost 100% or 98 to 100% of the hemoglobin gets saturated with with oxygen. So this axis here represents to you how much hemoglobin is there saturated with oxygen, or how much oxyhemoglobin is there. So the more the oxygen is there, the more the oxyhemoglobin formation will be. Then there are three curves here. The central curve here is the blue curve, which is the normal condition. The one which is above and one which is below, they both refer to a low CO2 in the tissue and a high CO2 in the tissue. So a low CO2 means a high pH, and a high CO2 actually means a low pH. So let's suppose in normal condition, the tissue already had consumed this much of oxygen, and the tissue had 30 millimeter of mercury of oxygen left. So if the blood is passing through this tissue, we'll look at the normal curve. We just draw a line here, and we touch this curve here, and we drop this value to the y-axis to read off the value from here, and we see that about 45% of oxyhemoglobin lost the oxygen to the tissue. So this much of oxygen was given to the tissue, so this hemoglobin lost all this oxygen to the tissue, which already had 30 millimeters of mercury and was respiring at a normal rate. Now let's suppose if the tissue was not this tissue but the one which is depicted in the green line, what do we see here? We see that in this case 32% of oxygen moved to the tissue. So why still if you see this value is 32%, so this line shows to you that 32% of hemoglobin lost its oxygen, whereas to this tissue 45% of oxygen was delivered to the tissue. So if you compare the value of 32% versus 45%, you see that the tissue which had a normal carbon dioxide received more oxygenated blood, and the tissue which had a lower carbon dioxide concentration, it received less oxygenated blood or less oxygen from the blood. The same goes for a tissue which is having a high concentration of carbon dioxide. Here the argument will go reverse here. So let's suppose if the carbon dioxide concentration in the tissue is higher, so we'll look at the red curve here, this line. So if that tissue also had 30 millimeter of oxygen left, so again we'll just move here, we'll touch this graph here, we'll read off the value from the y-axis. We see that this tells us that almost additionally this much amount of additional oxygen is given to a tissue where a high CO2 exists. So this actually gives us the value of about 48%. So 48% of oxygen was given to the tissue which had a high CO2, and 45% was given to a tissue which had normal CO2, and 32% was given to the tissue which had a less carbon dioxide. This can be explained on the basis of Bohr's effect, because when Bohr's effect is there, you have a high carbon dioxide concentration, and that high carbon dioxide concentration cause more formation of HHB, and when HHB forms, hemoglobin ends up losing more oxygen to that tissue, and the curves shifts towards a bit lower than the normal. In cases of low CO2 concentration in the tissue, the same normal curve goes a bit higher. Temperature and the accumulation of DPG also have effect on shifting this curve. So a high concentration of DPG and a high temperature shifts the curves lower, and a low DPG and low temperature shifts the curve a bit higher. This sums up what we have just discussed. The carbon dioxide from the capillary wall, it diffuses, it moves into the plasma. From the plasma, it comes into the red blood cell. Inside the red blood cell, some of the carbon dioxide forms dissolves and combined with water to form H2CO3. This reaction is catalyzed by the enzyme carbonic anhydrase. H2CO3 will dissociate into HCO3 ion and H plus ion. The HCO3 minus ion goes out and chloride ion comes in. This is called chloride shift. H plus ion binds with hemoglobin to form HHB. Hemoglobin, which was already having oxygen, cannot keep the oxygen attached once HHB is formed, so it allows some of the oxygen to be left out. So this oxygen diffuses out of the red blood cell and goes into the goes outside into the tissue. So this is how the gas transport or oxygen transport is affected by not only the diffusion gradient but by carbon dioxide concentration as well. We know that the acidic and the alkaline conditions can be found. We need to keep these markers in mind. If the pH drops below 7.35, the body goes in acidosis. If the pH rises above 7.45, the body goes in alkalosis. PCO2 is how much carbon dioxide is present in the body. So if it goes above 45, so the body is going in acidosis. If it's going below 35, the body is going in alkalosis. Anything in between is normal. Similarly, bicarbonate ion, if the value drops down below 22, the body will go into acidosis, and if the value rises above 26, the body will go into alkalosis. PCO2, since carbon dioxide is moved out from the body through the lungs, so this is a respiratory marker, whereas bicarbonate is a metabolic marker because it goes out mainly through the kidneys, but yes, to a certain extent in your respiratory system as well. So the bicarbonate for alkalosis and acidosis is considered to be the metabolic marker, and CO2 is considered to be the respiratory marker for acidosis and alkalosis. The values in between these are the normal values. So these are the few cases in which we see that if the pH of the if subject's pH is 7.79 and CO2 is 24 and bicarbonate is 21, we see that the pH is in the basic condition, and the carbon dioxide value given to us here is 24, so that's again in basic condition. So this individual is suffering from respiratory alkalosis because the pH is shifting to the basic, and the carbon dioxide concentration is also going to the basic region. And since CO2 is the marker which is being shifted, so that's the respiratory condition and this is alkalosis. But the metabolic marker, which is bicarbonate, this is 21, and this is lying in the acidic condition, so that's metabolic compensation going on. The same thing can be applied in another case where pH is 7.17, and this pH can be compared against the bicarbonate and CO2. So we see that the pH and HCO3 are in the acidic condition, the CO2 is in neutral condition, so there is no compensation here, but metabolic acidosis is occurred. All these conditions can be seen by this CAB buffer line. The CAB buffer line shows you the effect of adding bicarbonate to blood. So when you add bicarbonate to blood, the BAC or CAB buffer line actually can shift because of the bicarbonate being added or being removed from the blood. This is called as a Davenport diagram. The Davenport diagram shows to you the shift in the pH around 7.4. Above 7.4, it's alkalosis till if you keep on going ahead, and dropping below 7.4 is acidosis. This is the plasma bicarbonate concentration. All these points in this Davenport diagram are showing to you the buffer lines. The BAC buffer line is where we lie, so this is the normal condition. So this shows the relationship between pH, bicarbonate, and PCO2. So this region, this shift in this region actually shows to you the excess of base. So this part of the graph shows to you the excess of base, and this shows to you that somehow the body is having an excess of alkaline markers, whereas this condition dropping down below the BAC line shows to you a base deficit or negative base excess. Capillaries, when they are providing blood to the tissue, the blood is being provided by capillaries which are present around the tissue. So, for example, if this is a muscle here at the center receiving capillary oxygenated blood through the capillary at this point, so this distance actually tells you that as the distance decreases, the oxygen supply can also be given at a faster rate. Here you can see that the distance is increased. So if the distance is increased, yes, oxygen will be still provided, but it will be not very efficient. In the case of the gap being higher than normal, which means that the tissue is here and the distance between the tissue and the capillary is large, then oxygenated blood supply to the tissue will be affected in such a way that enough oxygen cannot be supplied to the tissue for aerobic respiration to continue. So the distance between adjacent capillaries having the tissue in the center should be minimum to allow a quick exchange. That's it from my side regarding the gas transport by blood. Thank you so much.